thank you everyone for joining us for this amazing afternoon of talks. Our last speaker is Dr. Gussie Maccracken. Dr. Maccracken is a National Science Foundation postdoctoral fellow at the Denver Museum of Nature and Science. And not only will share with us what fossil leaves can tell us about the plants that are in an ecosystem with dinosaurs, but also about some of the really tiny creatures that there are as well. Take it away.
Thank you, everybody, for sticking around this late in the day. So today, I'm not going to be talking much about dinosaurs. What I want to do is color in the rest of the landscape for you, or at least parts of the landscape for you. So when, let's see. Okay. So when you think of the age of dinosaurs, and you ask yourself, who are the leaf eaters? Who are eating plants? You probably are thinking of these charismatic dinosaurian megafauna, like this beautiful ceratopsian and this hadrosaur. But I'm going to ask you to sort of extend your mind and think a little bit about insects today. And this is because insects are the most diverse animals on Earth, and they also make up the biggest biomass. They're the most abundant animals in most ecosystems or land-based ecosystems. So if you see here, there's a pie graph showing the total number of described species on Earth today, and insects make up more than half of that. And then when you think about the herbivorous insects, those insects that eat plants, that makes up about half of the insect species we know. So between herbivorous insects and the vascular plants, that's about half of our described species, which is a huge number. I mean, it totally is overwhelming when you think about the small number of things with backbones like us. So one of the problems I have with insects is that they don't fossilize very well. So, you know, it's kind of a big bummer for me because I like to think about plants and insects. So what do you do when you don't have insect body fossils? But insects are generally preserved in a number of different ways. Here we have a Lagerstätten, a deposit of extraordinary preservation. Think of it similar to like the Green River Formation in that's Eocene in age. So an insect, in order to be fossilized here, falls into the water, it floats to the bottom, and it gets buried very rapidly so that it's not decomposing. And they're also delicious to things like, you know, fish. So you want it buried quickly, and you want it buried with very fine sediments because insects are tiny things for the most part, right? So you need that fine grain in order to preserve the detail of these fossils. Now we also have preservation in amber, but both these Lagerstätten and these amber deposits are just so patchy through time and space. They really, you know, when you get them great, you can get a great snapshot of the insect fauna of that area at that time. But a lot of places that we work, we really don't find many insects preserved, or sometimes any. So what do we do when we can't find insect body fossils, but we want to know what the most abundant animals in an ecosystem we're doing? We look at their trace fossils, and I study the trace fossils of insects on fossil leaves, and then sometimes things like dinosaur bones. But today we're going to talk about fossil leaves. So just like today, you can find holes and galls and all sorts of things that are preserved with the leaf. And this goes back about 400 million years. So you can really beautifully track these trace fossils through time. So what can insect damage on fossil leaves teach us about ancient environments? Well, we can think about the evolution of particular groups of insects and particular groups of plants. So for instance, in this talk, I'm going to be talking a little bit more about the evolution of flowering plants called angiosperms, which are a very recent, geologically speaking at least, very recent development in Earth's history. And we can kind of figure out how angiosperms evolved and then how insect populations fed on those and changed through time. We can think about how plant and insect communities adapt to climate change. For instance, during periods of really high global temperature, when the carbon dioxide is much higher in the atmosphere, it actually influences how insects have to feed. So plants have a ratio of carbon to nitrogen. Nitrogen is more limiting and insects need it to survive. And so basically the amount of carbon dioxide in the atmosphere changes that ratio of carbon to nitrogen, and insects have to feed. They basically have to eat more plants to get that same level of nutrition. So you can see during periods of warmth that we find that more of the forest is actually consumed by these insect herbivores. We can look at extinction events and recovery. So that's what I'm doing now for my postdoc at the Denver Museum. I'm actually looking at plant and insect communities in the Cretaceous, and then right after the mass extinction event that killed off the non-avian dinosaurs. And then what we're able to do is actually really record the tempo of recovery and how many tens of thousands of years it took for things to show back up in these landscapes. And then finally, we can think about the evolution of modern ecosystems. Like, why do we see the biodiversity of plants and insects that we see today? And where do we see them on Earth? And so we try to track those in deep time. So I just want to show you sort of the bedrock of some of my most recent work. And it's based on thinking about how our modern world came about. And this modern world is mostly a flowering plant dominated modern world. About 95% of all plant species are flowering plants. In fact, one of the things that I love in, like dinner party, like chit chat is, humans eat mostly flowering plants, and then asking people, well, what do you think you eat that isn't a flowering plant? And so people are like, oh, corn, well, that's a flowering plant. Mushrooms, well, that's not a plant. But the answers are things like pine nuts. And then one of my favorites is gin, because it has juniper berries in it. So most of what we eat are flowering plants, which really kind of sets the stage for thinking about what the earth looked like when they didn't exist yet. So we see that in the early Cretaceous, before flowering plants had evolved, we have things like ferns. We have things like other sea plants like ginkgo or neophytes, if you've ever been to Namibia. We have cycads, and we have conifers for the most part. But then around 136 million years ago is when we find the first flowering plant fossils. And after that, the rise in radiation of flowering plants is unprecedented on Earth. No other group of organisms has risen to dominate the Earth quite in the same way that flowering plants have. So by the late Cretaceous, they make up the bulk of species, or at least the biggest percentage of species of plants on Earth. And then it continues through to today. Oops, sorry about that. So my mostly dissertation research, and then after, I've been working on fossils from here in Utah, from the Grand Staircase Escalante National Monument in the Kuiperowitz Formation, which Dr. Lowen mentioned a few times today. So hopefully this kind of fills you in on some of the other portions of those ancient ecosystems that you might not have thought about. So here's what North America looked like about 75 million years ago. The climate was a bit different than today. It was a bit warmer. And so sea levels were higher, and the middle of North America flooded in what we call the Western Interior Seaway. And where we are in Utah would have been kind of in this ancient chain of mountains, and the Kuiperowitz Formation, which is south central Utah, was along this beautiful alluvial plain. And so here's what the Kuiperowitz looks like today. Kind of stark, beautiful, expansive badlands. But here's what we think it looked like in the past, 75 million years ago. As Mark said earlier, kind of think of it like the bayous in Louisiana, just these big meandering rivers, totally bordered by these newly evolved angiosperms. And so to collect data on the interactions between plants and insects, I'm going to just walk you through a little bit of what I do. So each type of damage gets its own number. So we can track the diversity of damage. And this is just based on the type of damage, the size, the extent, the shape, things like that. We have over 400 different kinds of fossil damage today. And then they're grouped into these essentially feeding guilds. We call them functional feeding groups. And I'm going to go through a few of them. We have hole feeding, when an insect chews a hole into a leaf, pretty self-explanatory. We have margin feeding, the insect chews the edge of the leaf. We have skeletonization. This makes kind of a pretty lace pattern. The insect is chewing a hole into a leaf, but avoiding the veins because they're tougher, it might not be able to chew through them, and sometimes they have defensive chemicals that can be released if the insect kind of snips that vein. Because remember, plants are alive. They're not just sitting there waiting to be eaten. They are producing really nasty chemicals. And I can talk about those after the talk if you're interested. We have surface feeding. This is when the insect chews off just one surface of the leaf. Sometimes it's the underneath side so that something, in the treetops won't be able to see that there's an insect, chewing on that leaf. So it's kind of hiding. We have piercing and sucking. This is done by an insect that doesn't have a chewing mouth part like the ones that I just showed you. It has a mosquito-like mouth part. So it's actually piercing into the leaf and then sucking out the good juices and cells and all that wonderful stuff. We have galls that we can find in the fossil record. I'm sure you guys have seen galls at some point in your life on modern leaves. This is when an insect lays an egg into a leaf. The egg hatches into the larva, and that larva will produce oftentimes a hormone that mimics the plant growth hormone. So it kind of tricks the plant into building its own little house. And sometimes it's a food supply too. Very sneaky. And then we have my favorite type of insect damage to find on fossil leaves. This is leaf mining. So again, an insect lays egg into leaf, egg hatches into larva, but then the larva doesn't stay put. It starts eating its way through the leaf. And oftentimes the shapes that it makes and the size of the leaf mine tell us what kind of insect made it today. So you can actually see, I don't know if I have a, yes, okay. So you can actually see that the oviposition point is right here, and it starts to wind its way outward. And as the insect eats, it gets bigger, and that the width of the mine actually increases. So you can actually see how this insect was moving 75 million years ago. And then finally, we have oviposition, or egg laying on the surface of the leaf. Now, the thing about what I do is that it's not an exact science. So one insect is capable of making, or can be capable of making, several different kinds of damage. And then conversely, several kinds of insects may be capable of making the same kind of damage. So it's not really a one-to-one. We have 30 types of damage. We have 30 insect herbivores. But it does give us a way to compare sites through time. And then if you're very lucky, sometimes you find a type of damage that is identical and distinct enough to modern damage where you can tell what kind of insect made that. So very, very briefly, I'm just going to go through some of my more recent discoveries. The first one is a new leaf miner from the Kyparoids, so it's 75 million years old. And so I'm sitting there, I'm looking through 4,000 and some leaves from a single locality a few years ago, and I find this kind of broken off partial leaf mine. And I said to myself, like, I have not seen that, and I've looked at so many leaves over so many years. And so I did what every good scientist does, and I Google searched it. And I found a leaf mine that was almost identical, because it's a really weird one. The insect starts in the middle and then just does a perfect little bullseye spiral all the way out, leaving this frail trail of frass, which is what entomologists call insect poop. And so I found out that the insect responsible for this leaf mine was called Leafcutters. It's a genus of moths. Here we have the adult at top and the caterpillar at the bottom. And so what I did was just described this ichnogenus. So the same way, you describe a great dinosaur, I can describe a fossil leaf mine if it's really cool, if it's really interesting, and especially if I can link the insect culprit that made it to a modern insect. So we called it Leucopteropsa, meaning Leucoptera-like, spirale, because it spirals. It's very complicated stuff, I know. And the importance of this is now we have a taxonomic identification of an herbivore from the Kuiperowitz formation. And also, we were able to basically find the only evidence of this group of moths in the fossil record. We don't have any of their body fossils. We just have this one single dumb little leaf mine. All right, I'm not going to talk too much more about insect damage, but I just wanted to show you what else we can do with this kind of data. So we can look at all the different kinds of plants in a locality. So here's a plate that I made of all the different angiosperms that I find in one of the localities in the Kuiperowitz. And then we can do this thing called rarefaction, where we basically build curves for the amount of surface area that's removed from a particular type of plant, and then look at the number of damage types. And this helps us compare how intensively insects are eating one type of plant versus another. So you see this kind of purple, kind of arch at the top. That is actually the fossil L, and it has a lot of insect damage. This thing was chewed to bits. Insects must have loved it. And then we have this kind of lighter blue rarefaction curve, and this is actually fossil D. And so what this is telling me is that insects have preference for these fossils. Some are being attacked more often than others, and this can really tell us also that maybe one has great chemical defenses and the other doesn't, or there's this tight coevolutionary relationship between certain plants and certain insects. Okay, I'm going to go quickly because I know we all want to get home, but my last little short description of recent research is finding the oldest plant mite mutualisms. So what's a mutualism? It's a cooperative association among species. The big two plant animal mutualisms, I'm sure you've all heard of them. Pollination, number one, huge, right? Seed dispersal, also pretty, important. And scientists think that these two mutualisms were probably very helpful in the radiation of angiosperms in the Cretaceous. So one of the reasons why angiosperms might have been so good at outcompeting other types of plants. But the third one is a type of mutualism I would be shocked if anyone here was familiar with, called a keratimatia. So domatia, domicile, means house. Acaro means mite. These are structures in the vein axils of leaves, modern and fossil, where basically the mite gets to live in exchange for protecting the plant. And it's the third most common type of plant-animal mutualism. So they form these beautiful tritrophic associations where the plant provides this nice house for the mite so that it doesn't get eaten by something else, so that it doesn't dry out. The mite then protects the plant by eating fungus that grows on the leaf, or depending on what kind of mite you are, you might eat other herbivorous mites that attack the plant. And this is what we think that these Kuiperowitz keratimatia might have looked like. They, you just see this little pocket, mites living in there, easy peasy. And so the most interesting thing, I think, that we found from this was not that like, oh great, mites live in, plant pockets 75 million years ago. I mean, kind of who cares, right? But we can look at the timeline of evolution and make some kind of speculative arm wavy sorts of inferences. So like I said earlier, first fossil angiosperms found around 136 million years ago. We also have the first kind of radiation of angiosperms that would be acceptable to produce a keratimatia around 100 million years ago. The previous oldest known keratimatia was from the Paleogene. We will just ignore that. And then we have ours at 75 million years ago. So what we did was extend the record of this mutualism back from the Cenozoic to the Mesozoic, 25 million years. And we also, were able to sort of narrow down when we think it might have evolved, probably around 100 million years ago. And so if we go back to this chart of percentage of species versus age, one of the things that makes angiosperms great is that they have all these amazing plant animal mutualisms. And so we're throwing a keratimatia into the ring as potentially one of the reasons why angiosperms, were able to radiate so quickly in the Cretaceous and outcompete everything. All right, so insect race fossils in the Herbivorous formation provide some of the only evidence of insects from this formation. I have found like one dragonfly wing, and that's it. And I have spent so long out there. We can see a kind of a baseline for late Cretaceous plant insect associations. We have the first occurrence of that Leucoptera leaf miner, and we also better understand the evolution of mutualisms during that early kind of rise in radiation of flowering plants. So with that, I just want to thank all of my collaborators and institutions and funding agencies for helping me along the way. And I would love to take any questions you all may have.
Let me articulate this correctly. On a micro level, I guess you could say, has the slow fall of like pollinating insects had an effect on the number of flowering plants versus say ferns or whatnot in recent years, of course, but we're seeing more ferns and less flowering plants?
Yeah, so ferns are kind of an interesting story. There is some evidence to suggest that they even diversified a bit after the radiation of flowering plants. But yeah, one of the things that does make flowering plants able to really compete well, to reproduce well, and therefore outcompete other groups of plants is that they have things like pollinators. Because if you don't have an animal pollinating you, then you're relying on things like wind dispersal for your pollen. But a pollinator actually targets certain kinds of plants and then targets the same kinds of plants for the next time when they fly off and fly to a new plant. So you really get, you waste less pollen. You don't have to spend as much energy on that. So that's one of the reasons why they, might be better than, not better. That's kind of biased of mine. They might be, more able to compete, to outcompete things like ferns. And they also have a different growth regime. So they're kind of weedy. They can kind of just crop up in very disturbed areas and outcompete slower growing things like conifers. Did I answer your question, Milo? Okay, sorry.
So my understanding is that insects were some of the first living creatures to develop flight. Do we think insects developed flight to coexist with plants, or do we think that developed more on the grounds of like traveling and moving themselves around and things like that?
I would say traveling and things like avoiding predators, because before insects were herbivores, they were detritivores. They can climb up and down these plants. They can get everywhere they need to be, basically without flight. But flight is great for moving, for finding mates, for escaping things? It's a great question.
I was wondering if you were able to infer things like oxygen content from one era to another kind of thing, like based on the number of insects, the plants and things. Like, can we tell what the atmosphere and the environment was like based on your research?
Yeah. So maybe not necessarily oxygen levels, but with carbon dioxide, we oftentimes look at the margins of leaves, and there's essentially an equation that tells you the proportion of leaves with smooth margins to the proportion of leaves with toothed or serrated margins follows a pretty straight path. And so we can actually predict the temperature of different sites and also sometimes the precipitation. And that gives you an idea of carbon dioxide levels in the atmosphere. Now with oxygen, one of the cool things is that insects don't have lungs like we do. They have a different system of breathing. And so they're kind of limited in their size by the levels of oxygen in the atmosphere. But in periods of time when we have much higher levels of oxygen, like the Carboniferous, we actually see that insects get really big. So maybe you've seen those enormous 3-foot wingspan dragonfly fossils. So that occurred because we know that oxygen was higher. But one of the greatest things about my job is that we work with a bunch of different kinds of paleontologists. Our field is very collaborative these days. So we work with geochemists and people that can help us get to these kind of atmospheric types of questions. We work with stratigraphers and geologists. We work with modern ecologists, and then we package that all up, and we can make some really cool hypotheses about what was going on in deep time.
Got a question here.
So with the notoriously bad insect fossil record, how difficult is it to figure out what their role was in past ecosystems? We talked about how insects are so important today. How confident can we be that they were that important in the past as well, with how bad the fossil record is?
That's a great question. So like all paleontology, it's based on observations of the modern and just inferences. But for at least my work on herbivory on fossil leaves, we see even when there aren't body fossils, entire forests can be basically decimated by insect herbivores. And also insects haven't really changed too much over the past few 100 million years. They're really good at what they do, and their body plan is really stable. They can make it through anything. And so if you were to look at an environment, say 200 million years ago, and you're just kind of squint and look at the fossil or the insects of that time, I don't know that you'd recognize that they're different species in many instances. Obviously, some things go extinct and species turnover is always very high, but their mouth parts are really adaptable, but they kind of stay within a range of body plans, and the body plans really tell us what they do. Yes.
Well, this is kind of a similar question and feeds off of what you just said, but it occurred to me that if the angiosperms took off in that way in the late Cretaceous and underwent a huge evolution, that the insects might have undergone an evolution as well. And I wondered, do you see any changes in the feeding patterns on angiosperm leaves versus the earlier fern leaves that might indicate that?
There definitely are differences in feeding. We know that there are radiations of insect, clades that coincide with the evolution of flowering plants. But it's still kind of hard to pinpoint that, really. I think what we are trying to do more is look at molecular phylogenies and figuring out when things evolved and if that really coincides with the evolution of flowering plants. So like there's some kinds of beetles that probably radiated alongside flowering plants. And then when you have your pollinators, your butterflies, your bees, your, things like that, then we expect, and we do in some cases see that they, really radiated. But there's also switching going on. So there are lineages of insects that we see feeding on conifers, and then we think they just switch to angiosperms, and there's some that just stay on conifers. And so it's all over the place. But, that's one of the things that we've been really trying to pin down over the last 10 years or so. But this field is still pretty relatively new compared to a lot of other parts of paleontology. So there are a lot of big questions that still need more data behind them, I would say.
We have a couple, if you're willing to take a couple more questions. Randy, it's Randy over here. We have a few questions from online.
Yeah.
First one is there any evidence that these mites and other insects were larger or smaller in the late Cretaceous? And does that change the size of the damage they leave?
Yeah, that's a hard question to answer, but a great question. So, we think that mites probably, they actually have a huge size range. So some of the mites living right now in your eyebrows, you can't really see them, but some mites you can see with the naked eye. So there's always that size range. We think they were probably about the same size. Same thing with insects, but they're orders of magnitude of difference between different groups of insects. So that's a question I don't think we can answer with a lot of confidence. But I think the, at least the oxygen content one wasn't going to allow for things like those giant dragonflies.
And the other question, which is a really good one, is when you showed that graph of different plant groups through time with the radiation of angiosperms, did angiosperms just get more diverse, or when they got more diverse, did other plant groups decline in diversity as well?
Yes, the answer is yes. So angiosperms become very, very diverse very quickly. And we do see that there is a decline in the number of things like, especially cycads. Like in the early Cretaceous, you'd be bumping into cycads everywhere around here. But then, by today, these are, still pretty much angiosperm dominated ecosystems if you look out the window. And today, you're not going to see a cycad for many, many, many miles. But yeah, so conifers, cycads, even ferns, probably their numbers probably dropped because they couldn't compete as well with angiosperms. But there is a little bit of, there is some, I guess, strong evidence that ferns also had their own radiation in the shadow of angiosperms.
Yeah, we have time for one last question.
This is probably wishful hoping and thinking on my part. Any evidence of leafcutter hymenopterans?
Yes, I believe did I delete that slide from my talk? Okay. There you go, right? So leafcutter bees, there's evidence of leafcutter bees in Germany. Actually, we have evidence all over the world by the Eocene. So it's one of the only ones that is specific enough to really determine like this insect made it. And in fact, I saw some leafcutter evidence when I was down in the tropics a couple of years ago, and I lost my crap. But there are other great like types of insect damage that you can really tell what they are, especially with leaf mines, because there are 4 main types of insects that make them today. And so, sometimes we can find the culprits and we can make good taxonomic, identifications, at least at an order level or a family level. And then in my case, I got to do genus level. So thank you all very much.
All right, thank you.